Hello, it's an honor to be speaking for the second time to the Philosophical Society. The first time was just over 10 years ago when I presented our research on the origins of humans, how humans emerged from Africa 60,000 years ago to colonize the planet, continent after continent. And my talk today is on the origins of the coronavirus. And those two talks are related because in both cases, for humans and now for the virus, I use a technique, phylogenetic network analysis, with which we successfully found the origins of humans. And as I hope to show you, we will trace the origins of the coronavirus as well. So my work on human origins was starting in the 1990s. I teamed up with a mathematics professor and we developed this phylogenetic network technique to apply it to human DNA variation. And that was difficult previously because researchers had used tree algorithms instead of network algorithms. And allow me to explain in this talk what the difference is and what the advantages are for using networks. So the work on coronavirus started in my mind in January 2020 when I noticed that the reports emerging from China indicated a quite lethal virus, and I thought we'd be in trouble if it manages to escape out of China into the rest of the world. So I decided it was time to trace the evolution of this virus as quickly as possible, and that means there isn't any time to apply for research grants. There's no time to put a team together of outsiders. So instead, I recruited my old Professor Renfrew, who started the archaeogenetics group in Cambridge Archaeology. And I recruited my wife, who's a practicing medical doctor, but has experience with DNA analysis, and my brother Michael, who works in cancer genomics and therefore also has the tools required for this coronavirus project. So the first genome, so here you see the PNAS paper we published early last year. I believe we were the first Western group to publish the evolution of this virus. The timeline is on the right of this slide. So on Christmas Eve 2019, the first coronavirus genome was submitted to an international database by Chinese scientists. We collected data from this database with a cutoff date until early March. These were 160 high-quality viral genomes. Then we spent 2 weeks on analysis and preparing the manuscript and submitting it. We had 2 rounds of reviewing, referees who criticized and commented and required changes. And by the time we implemented all that, it was April the 8th when we published. The coronavirus data comes from an international database called GISAID, and this started off in 2008 as an influenza database based in Germany. It was a private venture which after a few years was then supported by the German government. The database then switched to accepting coronavirus genomes when it became clear that this epidemic was going to spread. And I'm indebted to those who have set up this GISAID initiative because without all this preparation over many years, we would now not be in a position to analyze this virus so quickly. And I compare this to, for example, the Jenner Institute in Oxford, which had existed and had researched on coronaviruses for many years and therefore was in a position to help develop vaccines when the time came. And in a similar sense, I suppose our network analysis, which was scheduled for the 1990s work on human origins, now became immediately available for analyzing the coronavirus. Now, what are these networks? Let me show you with this simple diagram. So imagine you have a virus genome some time ago. It would have started off with a single virus spreading in a species such as a human. So a single genome of a virus started off, and this is demonstrated in this circle on the top left. The small red circle, top left, is located somewhere in space and time on this planet. If this virus is successful at infecting people, it will do so, and now more people will carry exactly this virus genome, and that is indicated here in the second step with a larger circle. So these are many virus genomes in different people. Now in the third step, you will start seeing the effect of the DNA or RNA mutation rate. So we have in this star-like cluster, which at this point is a tree, we have at, let's say, 2 o'clock, position 16,303, which is now leading to a new coronavirus variant or mutant. So the original red ancestral coronavirus type is becoming more and more rare over time because more and more mutations are happening, creating new types, which I show here as these white circles. The coronavirus genome is 30,000 nucleotides long. And if I say here at this 2 o'clock mutation 16,303, that might be a change from, for example, A to G. Or if you look at 9 o'clock, you have the position 13,068. That might be a mutation from T to C, creating this new variant shown in the white circle. So the important point here is that it's no longer the frequency of the ancestral red type, which shows you there was a successful viral expansion event generations ago, but it's the entire star-like tree cluster, which shows you that a virus successfully infected some time ago. So let's move forward to the final step. Now we run into technical problems. So first of all, more and more mutations are happening. The branches are getting longer and longer. And in fact, the red ancestral type might now become very rare or become extinct, so you don't have it in the sample you took from the patients. So that means you have to reconstruct it. And this requires something called phylogenetic analysis. That is the reconstruction of the ancestral types based on the existing white types you see in this diagram. Now, if you have a genome which is even 30,000 nucleotides long, there is always a possibility that the same position will mutate several times independently in different branches of this evolutionary tree. And that causes problems in the sense that you now generate a data set which could produce, in theory, different tree solutions. So take a look at this final step. You have the red ancestral type in the center, which is nearly extinct. You have coming off at about 1 or 2 o'clock a position 16,208. And from that, you have another mutation 16,303. And then you have a cycle that is a diamond, which has these same positions on opposite links. And this diamond, so that's more or less a squashed rectangle, incorporates 4 potential trees, because I can take away each of these links in turn, and then I'm left with a tree solution. So one diamond is 4 trees. Now attached to this diamond is another diamond below at about 3 o'clock, and that's another 4 trees. 4 trees times 4 trees minus this common link and the domino is 15 trees. And if you look below at 4 o'clock, there's another diamond, and that's another 4 trees. So 4 times 15, that's 60 trees. So you have 60 trees in one network diagram, and that's all available at one glance, and you can immediately and easily see what's going on. If I showed you all these 60 tree solutions on this PowerPoint slide lined up, you would not see the wood for trees. And this is the reason why our network analysis was able to uncover human origins, because we could make sense of the data at a glance. Whereas if you tried looking through large numbers of trees, you wouldn't get anywhere. So applying this phylogenetic network analysis to our coronavirus data set, which is the first 3 months of the data being published until early March, we find that the network, which you can see here, is fairly easily intelligible. You have these fat star-like nodes which are linked together. We've called them A, B, and C. And you see that the colors here reflect geography. So the yellow and the orange colors mean these central nodes, these ancestral nodes, are found in China and in Asia. And then you can see largely in the more peripheral nodes these blue colors, and these are Western populations, so Australia, North America, and Europe. And the first thing you'd like to do with such a network is to find out where is the ancestral type. And in order to find that out, in genetics you take what is called an outgroup. You take a species which is outside your own species, so you're sure, you know, it is disconnected in that sense from you, so it's distant in time. And the obvious choice is to take the bat coronavirus, which is 96.2% identical to the human coronavirus, and can therefore point within the human data set, within the human tree, which is the ancestral node. And as you can see, the bat attaches to this cluster which we have called A, so that is the ancestral type that first started spreading in humans. So now we come to the actual topic of this lecture, that is the origins of the current coronavirus pandemic. So I would wish to distinguish three different levels of origin. At the first level, let's take a look at the species jump from the host animal to the human host. And you have heard various theories, probably from the media. Was the bat indeed the host for the first coronavirus infection in humans, or was it the pangolin, or a mix of the two? Did this infection happen in 2019, or did it happen much earlier? So these are the questions we need to look at. Then the second level of origin is the successful spread of the pathogenic virus in Asia. Now, why do I distinguish these two levels? And I do so because imagine that the bat infected a human many years ago, a long time ago, but with an asymptomatic strain. That means humans carry this virus around with them, but it was never noticed, it never caused any symptoms, so no one analyzed them. And it was only at a much later date, in this hypothetical scenario, that the human virus acquired mutations which made the virus pathogenic and therefore caught the attentions of the medical profession, and therefore the virus was first discovered. So that is what we're looking at in this second level of origin, the emergence and spread of a pathogenic variant in humans. And at the third level of the origins question, I want to take a look where the current global dominant strains come from. Now, as you know, we've until recently had the Kent variant as the dominant strain, or called also B117. Currently we have the Indian Delta variant, which is dominant. Doubtless there will be others, but these variants all go back, I will say this now, to a North Italian strain, which is a B strain, which I showed you in our network. And we'll take a look at those origins a bit more closely. Now, starting with level 1, the species jump from an animal to human. So in this picture you can see genome alignments. In the top rows you have the human coronavirus. Clearly the coronavirus is 30,000 nucleotides long, so this is just a small segment to demonstrate. Now below the humans you have the bat coronavirus in one row, and highlighted in the pinkish color you see the nucleotides where the bat coronavirus differs from the human coronavirus. And there are a few differences you can see in this alignment. Now below the bat we have several rows of pangolin virus, and you can see the pangolin coronavirus differs more substantially from humans than the bat does. And in the bottom two rows you can see another pangolin coronavirus strain, and that also differs substantially from humans. So the fact is that of all these potential hosts, the closest known coronavirus comes from a bat, and that similarity is 96.2%. Now, where does this particular bat come from? Well, this is an interesting story because in 2012, miners in China died of a mysterious pneumonia, and a research group decided to survey local bat caves to find out if viruses from bats could have caused these deaths. And so they surveyed bat populations from several caves in China, and then published a number of bat coronaviruses, including this particular one. And this publication was in 2013, included the famous Chinese researcher Zheng Li, working at the Wuhan Institute for Virology. And what is interesting is that since 2013, several years have passed, and other research groups have looked for other bats which might have a more similar coronavirus strain, and have not found a more similar one to date. So if we now take this difference of 4% between human and coronavirus genome and apply the mutation rate as a clock to find out when the common ancestor existed between the bat and human coronaviruses, this works out at about 50 years ago, so very roughly. This, of course, assumes that the mutation rate has stayed constant over time, which is not necessarily the case, but let's say as an assumption that's the best we can do at the moment. If indeed this age of 50 years between bat and human coronavirus divergence, if indeed that age means that the infection happened some 50 years ago, then clearly it must have been an asymptomatic infection, otherwise we would have heard about it before 2019. The other possibility, however, is that the human-bat transmission, the bat-human transmission, was much more recent, perhaps even in 2019, but we have not yet found the responsible bat population, and indeed this bat population might have gone extinct and we will never find it. So those are the two possibilities, but 50 years is the oldest feasible date for the bat-human transmission of the coronavirus that is now plaguing us. Now, my working hypothesis, and it's not more than a working hypothesis, is that any bat-human transmission cannot have been too many years ago, and that is because if the bat populations, if the coronavirus from the bat had many years to spread and disperse around the world, and then you have the pathogenic mutation which brings it to our attention, we would expect there to be a discrepancy between the origin, where the bats are, and then where you find the first clinical case. But in fact, there is no such discrepancy. So you do have that discrepancy, for example, in HIV and AIDS, where the cases were first clinically discovered in America, but obviously AIDS and HIV comes from Africa. You have no such discrepancy with this virus, and it's therefore likely that there was little time between the bat infection and the obvious clinical phenotype which is picked up. So how could this transmission have occurred? We've all heard of the Wuhan wet market or seafood market as a potential origin, but I believe the Wuhan seafood market is a red herring, because the first patient was diagnosed on the 1st of December, published in The Lancet shortly afterwards, 1st of December 2019, and this patient had no contact with the seafood market. I also have other reasons which I'll present later in the talk. What is the mechanism then of this transmission if it's not an animal on the seafood market? We don't know, but there is an indication from a study by my colleague Dr. Maureen Miller, who worked in the Wuhan Institute and is now at Columbia University. She had studied together with Professor Zheng Li, villagers in 2015 living near bat caves, and found out that 3% of these villagers were positive in antibody tests for bat coronavirus infection. So this was a related coronavirus, not the same one we have today. And these villagers have not provided, or this study has not yielded a clear reason why these villagers were infected. But we have to think of perhaps exotic possibilities, such as infection with associated with bat guano. So the guano from bats is a very popular fertilizer. It is mined, and it's even used as an eye treatment in traditional medicine. So there are potentials there for infection. Now, that was the bat to human transmission, the initial one, where I conclude it was probably a relatively recent event, even though the discrepancy is 4% or 50 years on the genome. We come to the second level of origin, which is the successful spread of the pathogenic virus, initially in Asia and then beyond. So here again is our published network of the first 160 high-quality coronavirus genomes. And we have our three types A, B, and C, and the root is in A. Now, at the time, that struck us as interesting or unexpected, because we assumed, like everyone else, that the virus had emerged initially in Wuhan. And it was surprising that Wuhan didn't have the ancestral A type, but the derived B type predominantly. We didn't make much of it in our paper because it wasn't controversial at the time. But now clearly there are various theories on where this initial spread started and how. Now let's take a look in this slide at the first 3 weeks of the known epidemic. And here I have plotted the virus genomes until January the 16th, as they were known in the international database. So we have 34 B types in total at this stage and 7 A types in this initial database. The A types are the black pie slices, and the sample sizes are clearly very small at this very early stage. But if I ask you where these A types are most common and where therefore is the probable area of origin, you would probably say, well, perhaps somewhere in China, Southeast Asia, but definitely not in Hubei province or Wuhan, because if you look at this, out of the 29 genomes available, only 1 is an A type. And elsewhere we have in China 12, and in Nepal we have 12 coronavirus genomes, and half of them are A types. So this doesn't really look as if Wuhan or Hubei are good candidates for the origin of the spread. If we now look a week later, we have 61 B types at this point, and then 22 A types. We can see that the A and B types have spread across China, and even Hubei is now becoming more similar to the rest of the China. We have 10 out of 48 A types. That's not quite as high as elsewhere, but it's now becoming more similar. What can be causing this spread? Well, I'm not sure, but I just note that January the 25th, 2020 was Chinese New Year, and the day before, January the 24th, is their New Year's Eve, where families travel to meet together just like they do for Christmas in Christian societies. And that possibly might have been a factor in spreading this virus initially, because people might want to avoid the rush and travel a little earlier. So, why is this ancestral type rare in Wuhan, and in fact in Hubei province, until the 16th of January? So our observations, I'll just repeat, are that there's only 1 in 29 Hubei patients who have A, whereas half of the 12 patients in the rest of China at this point have A. So these are not large numbers, but they're still significant. In fact, not a single A is found until the 5th of January in Wuhan, even though there's a sample size by now of 24. I have to add in parentheses, there is one more A in preliminary data which still needs to be geographically proofread. So this is a low number by the middle of January. And then after that, from the 17th of January onwards, 1 week before Chinese New Year, we have this assimilation where Wuhan becomes more normal in the data. Normal in the sense of having more A types just like in the rest of China. So the theoretical explanations, which I'll briefly go through, are, well, the sample sizes are so small they're unrepresentative. That's a possibility. I don't think that's the best explanation because some of this is significantly different. The second possibility is that there is selection going on. That means this B type is fitter, it's more efficient at spreading in humans, and therefore, in theory, A might have been originating in Wuhan, predominant there, but then B developed and took over and became the dominant type. So that's one possibility, entirely theoretical. I don't quite believe this possibility because we see 1 week later A's and B's spreading everywhere else in China. So why isn't B the predominant type elsewhere, just like in Wuhan? So to me that doesn't make sense, that theoretical explanation. The other possibility is sampling bias. For some reason, perhaps the doctors in Wuhan sampled predominantly B types and overlooked an existing pool of A types in Wuhan. Now I think that's also less likely because some of these Wuhan samples were indeed, or most of them, analyzed by local doctors in Wuhan, but half a dozen of these samples from Wuhan in fact were travelers who visited Wuhan, returned to their hometowns elsewhere in China and even as far away as Thailand, and the local doctors there analyzed them, found them to be positive and published their genomes, and they are also only B types. So I think it becomes difficult to defend a particular sampling bias given this situation. So we're left with the next possibility, which is a founder event. That is, the virus originated elsewhere, for example in southern China. The first person to arrive in Wuhan to spread the disease happened to have a B type. Therefore, this became the founding predominant type in Wuhan, which doctors then sampled. And it took a while for travel, for example, in the run-up to Chinese New Year, for other types then also to arrive in Wuhan, for example, these A types. So in total, this would make it less likely that Wuhan or Hubei province was the actual origin of the coronavirus. And I would like to note in this context that the previous coronavirus outbreaks, that is the SARS outbreaks in 2002 and the swine coronavirus outbreaks since 2016, they also were first reported in southern China, in Guangdong, for example. And this is where half our A samples are with the ancestral types today for the current virus. So it wouldn't be surprising if this third coronavirus outbreak also came from southern China. Now let's take a look at the time depth of when the coronavirus started spreading successfully as a pathogen in humans. And we created a larger data set for the time estimation. The cutoff date is the end of March 2020. And at this point, we have 1,001 high-quality coronavirus genomes from the GISAID database. And if we then perform this linear regression through the data points, we see that about 1.2, 1.5 mutations happen per month of the coronavirus, and then it accelerates a bit to about 2 mutations per month in February, March. And this early mutation rate we can then use to follow back when did the A-type virus have 0 mutations. And that is the same thing as saying when did the A-type virus first infect humans. The linear regression tells us this was at some point, so the first spread in humans was at some point between the 13th of September 2019 and the 7th of December 2019. So that's a 95% confidence interval. So probably around October, November is when the coronavirus pathogenically became obvious and was spreading in humans. And this is relevant because we often get the questions from the public saying, "Doctor, I had the symptoms in 2019. Have I had the virus?" And this question generally arises after media reports which try to get at the time point of initial transmission. So, for example, a research group in Harvard University took satellite images of hospital car parks in Wuhan and surroundings, and they noticed the car parks were getting busier in August 2019, and at the same time people were Googling for the symptoms in the internet. And they suggested perhaps this is an August 2019 initial outbreak. We've also had sewage analyses being published, so initially in Italian and Spanish sewage works archives, and some of the early dates there, for example in Spain, may indicate that the virus was in the sewage as early as March 2019. So our answer to these questions is, well, according to our linear regression, there's only a 5% probability that the coronavirus was spreading successfully in humans before mid-September 2019. But we have to be careful with this date because we are assuming that the mutation rate is constant when we use this linear regression. But clearly, we have already seen that the virus mutation rate has changed over time, and therefore there's no guarantee that it hasn't changed in previous months and years, and therefore our time estimate would be accordingly inaccurate. But it's the best we can do for the moment to say that between mid-September and early December 2019 is, as far as we know, the best estimate when the virus started spreading in humans. Now, coming to the third level of the virus origins, let's take a look at the origin of the global strains which are now plaguing us. And before I get to that, I just have to show you the technique of using genomic tracing to find out in general where someone has been infected by the coronavirus, how we can trace on an individual basis where someone was infected. So let's look at first the first Canadian case. So this Canadian patient had visited Wuhan and Guangdong and then returned to Canada and fell ill on the 27th of January 2020. And this Canadian patient is shown here as this blue node with the label Ontario. And if you look at our network and follow back one mutation, one link, it leads to an empty node indicated by this blue arrow. So this virus genome indicated by the blue arrow doesn't exist in the data set, but it is reconstructed on the basis that you have these two yellow circles which derive from it. One of these is from the city of Foshan and the other from the city of Shenzhen, and both are in Guangdong province. And this indicates that the ancestral virus genome, which is not present in the data, also is from Guangdong province. And that fits nicely with our Canadian, who also was visiting Guangdong on his travels. And the story doesn't stop there. You can see derived from the Ontario node is a node with 3 Californians. The blue circle here with California in it is larger because it symbolizes 3 patients have this precise virus genome. And also 2 mutations further on, you can see another Canadian who has been infected by this type. Another example, here is the first recorded Brazilian case. So this Brazilian patient had visited Lombardy in northern Italy before his diagnosis, and then he was reported ill after he returned home on the 25th of February 2020. And if we want to ask a question where this Brazilian got his infection from according to the network, you can see the Brazilian is here, bottom left, the green circle, and 3 mutations further up you can see he latches onto a node with 2 Italians in it, so that clearly fits with his travel history. If we then want to go further back and ask where did these Italians get their infection from, you can see the answer is ambiguous, because this is part of a reticulation where the Italian could have got it from this orange and yellow node, which has virus genomes from Singapore patients and Hong Kong and Japan, Alternatively, he could have got his infection from this node, which is from a Chinese Australian in Sydney, and from this cluster here. And that's the strength of the networks. It not only shows you where the answer is clear, but it also shows you where there are alternative answers when it's not clear. So, returning to the question of where our global strains are coming from, here is the history. We have down here in the network our A type. So this is the one that arose, perhaps somewhere in China, perhaps somewhere in southern China. 2 mutations further on, we have a B type. This is the predominant B type in Wuhan. In Wuhan, mutations occur, so there are some derived types. Some of these travel to Western countries and become these blue nodes. And in one case, a B type mutates at 3 positions and is found in an elderly couple in Wuhan, a husband and wife. And they travel to Shanghai in January 2020 and visit their daughter, who is an engineer working for a German engineering company. And she then leaves Shanghai for Munich and arrives there on January the 20th, 2020. And unknown to herself at the time, she has been infected by her parents visiting from Wuhan. So nearly asymptomatically, she has training sessions in the German company in Bavaria with some German colleagues there, and then leaves a few days later. On the plane, she already notices she's coming down with a virus, and upon arriving in Shanghai is examined and found to be positive, and she alerts her colleagues back in Bavaria that there is a risk of infection. So the Bavarian colleagues are put under quarantine, and it appears that the quarantine is successful. About a dozen colleagues are infected, but this doesn't seem to go any further. However, if you look at the network, you find that only 1 mutation distant from this Bavarian type, labeled Munich here, you have the first case in Lombardy in Italy. And this is a young man, 38 years old, who gets very badly ill, so he has to spend weeks in intensive care. And there is no indication where he got his infection from. The flights between Italy and China were suspended by the Italian government at the end of January 2020. So it's difficult, but perhaps not impossible, for the infection to have come directly from China. The network indicates, however, a possibility is that the quarantine was not successful in Bavaria. And from there, this Lombardy virus type now spreads across the world. We all remember these hospital scenes in Italy with the corridors and hospitals full of dying patients. So this is clearly an infectious type, and you can see there it generates the first Mexican case. So this is a Mexican who had visited northern Italy. It generates more cases in Germany. This German here had visited Milan and then took it back to Germany, and from there it quickly spreads worldwide and generates the Kent variant, the Indian variant, and these are all called B variants based on the network here. And specifically nowadays B1 variants. Now looking more closely at the infection cluster in Bavaria and Munich, on this publication you can see the Shanghai engineer as a blue bar, and here on the left is the timeline, January the 20th, when she arrived and was in touch with these 4 colleagues, and one of them is patient number 4. This is a woman in her 40s. She she was infected herself, and unknowingly, unwittingly, passed the infection on to patient 5, and the only contact they had was sitting back to back in the work canteen, and she passed the salt, and that was sufficient for the infection in this case. On January the 24th, she was only mildly symptomatic. She had the chills, and so decided to go for a skiing weekend with her partner, which involves a 3-hour journey to the Italian border, to an Austrian ski resort, which is in Tyrol. So you see here, this is the valley, which is 2,000 meters high, and the skiing slopes go up 2,500 meters. My family and I happen to know it quite well because we regularly spend skiing holidays there. So at this point she had fatigue, a blocked nose, and sinus congestion, but that didn't put her off skiing. The hotel she used has a cabin lift, and that seats up to 8 people. And it's very popular at weekends if the skiing weather is good, as it was at that weekend. The hotel is a dormitory style hostel, and so she was in one dormitory with 13 people. She then drove back on Sunday at the end of the weekend to Munich, and there she was informed about the coronavirus potential infection, and sure enough she was tested positive, whereas interestingly her partner was not. Another interesting aspect here is that she harbored 2 types of coronavirus in her body, a mutated form and the original form. The mutated form is in her throat, but not in her sputum. And that means if her coronavirus is the source of the Italian and therefore the worldwide infection, it would have started potentially from her lung, rather than from a different part of the airways, and potentially that can be explained whether you transmit a virus by coughing or by sneezing or by speaking. That might involve different source areas within the body, but these are tests which I'm sure can be done. Right, so that essentially is the results session. Now let's go on to some methodological questions, because what I've shown you is fairly clear cut, I hope. So these are hypotheses which can be further explored if there is willingness to do so. But why is it that it doesn't seem such a clear-cut set of hypotheses to other researchers? And as I mentioned, the differences are partly methodological. So you have our phylogenetic network methods, which we prefer, but you also have tree building methods. And that's not the only difference. You have methods that use distance data, and you have methods that use character data, and this yields different kinds of results as well. So what is distance data and what is character data? If I ask you the question, what is the difference between, let's say, English and German? You could take a list of 100 words, and you can see some words are exactly the same. The word for nest is exactly the same in English and in German. But the word for woman is different. In German you say Frau, in English you say woman. And the word for cloud is different. You say cloud, and in German it's Wolke. And the word for bone in English is bone, obviously. In German it's Knochen, which is related to the English word knuckle, but it's a different word for this context. So you can either express it, as I've done just now, as characters and say there are certain features or characters by which German is different from English, by the words for cloud, for bone, for woman, and so on. Or I can give it to you as a percentage and say, in this list of 100 words, German differs from English by 30%. And if I ask you which type of information is more useful, a distance data or character data, your answer must be, well, character data is more useful to the researcher because you can directly say which features are different. Whereas distance data, 12% or 30%, is a more general kind of information. And the methods that we use are not any network methods, but specifically in this graph you can see character data network methods. And we published 2 methods in the 1990s, the reduced median method, which is RM for short, and the median joining method, later on MJ for short. And these are the ones that provide you not only the multiple solutions in the networks, but they also directly tell you which mutations, which characters on the virus genome are mutating and giving rise to these new virus types and virus phenotypes. Now, with that in mind, let's take a look at what other researchers have been saying. So this is a group from Florida, and they took the same data set as we did, except they added some new sequences which emerged a few days later. For example, 5 new sequences from Dutch, Welsh, Italian patients. And they disputed our finding, and they said, well, it's not clear at all that this global spread pandemic comes from this Italian type, as is shown in your network, because when we put in these 5 additional sequences, it shows the resulting tree becomes completely unstable. It is uncertain whether the Italians and the Dutch and these new 5 sequences, they slot in here or over there. So these branches are statistically unstable, and therefore you can't use virus genomes to trace where someone got the infection from. So we emailed these colleagues and said, could you please provide us with these 5 new virus genomes from 5 patients? And yes, they emailed them to us. And the first thing we noticed was these new sequences were identical to each other. There was no difference in the virus genome. So this is something the Florida researchers had simply overlooked. And the reason is, I think, because their tree building method assumes that everybody's different, and therefore there are deep branches separating these identical genomes. And that makes it look as if they're different, but it's simply an artifact of the tree building method, which uses, incidentally, distance data. And the second thing that the Florida researchers haven't done or haven't noticed is that these 5 patients have travel histories, and in 4 of the 5 cases they had visited Italy a few days before falling ill. And so it makes a lot of sense that their virus genomes are identical to our Italian virus genome, and secondly, they had actually been to Italy and presumably got themselves infected there. So my conclusion here is, please use experience and common sense in addition to statistics. More tree problems here. So we have other colleagues here. This was published in Science last year, where they tried to trace the origin of the European and North American infections and come to different conclusions. Let's take a look at their tree. So you can see down here there is a patient called WA1, that stands for Washington 1, first patient in Washington State. And then up here we have another patient, Washington 2. And this tree makes it look as if Washington 1, Washington 2 are quite far apart and quite different. And now let's count the links separating the 2 patients. So that's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 mutations back to this root, and 11, 12, 13 up again to Washington 1. So it looks as if 13 mutations separate Washington 1 from Washington 2. However, if you look at the sequence alignment table here, you can see the 2 patients differ by only 3 mutations. That means most of these 13 mutations shown in the tree are artifacts, and we call them ghost links. And we published this back in 1995, that these ghost links happen with distance-based tree methods. And it's such a pity that 25 years later, these mistakes are still being made. Another problem is the routing itself. So you can have an unrooted tree or an unrooted network, and then you want to identify which is the oldest type in your network, the origin of the whole network or the whole tree. And I remember in the early 1990s that a group in Atlanta tried to trace the origin of humans. So they took mitochondrial DNA samples from Africans, from Americans, from Asians, and when they wanted to find out the root of this human tree, they took an Asian to identify an Asian mitochondrial DNA to identify the oldest position in their tree. And obviously that made it look as if Asia was the cradle of mankind, and the whole world came from Asia. And this, instead of taking an out group, like for example a chimpanzee or a Neanderthal or an orangutan, they had made the mistake of taking an in group, so taking a member of the data set or of the same species in order to find the root of the species. So you simply can't do that. Unfortunately, this is being done by some people working in virology. they are assuming that the oldest patient or one of the older patients, the first recorded cases, probably is close to the root, and therefore you can use patients early on in the pandemic in order to find the root. But this shows you can't. So here is our network again. This time the colors don't reflect geography, but they reflect the week of sampling. So this is week 0, week 1, week 2, and so forth in different colors. And you can see there is absolutely no clear connection between the week of sampling and the color and the position in the network. In fact, look, the oldest patient is brown, week 0, and is up here at about 11 o'clock. And the bat here is diagonally opposite, locating onto our ancestral A type. So outgroup grouping is something you can't do without. And here's another example where all these problems come together. This is from a German medical magazine where you can see this tree. So by now you're experts, so you can see this tree is full of ghost links, which are artifacts. And in this tree there's an attempt to find out where these global strains come from, these variants of concern as they're called. This is the B117 in orange, which is our Kent strain. This is another B1 strain. And down here we have our first German case, the Munich cluster I talked about earlier, with these pale green branches. And as you can see, the problem with this tree is that the branches do not link up with the Munich cluster, but they go straight back to the assumed root of this network, as if there's no genetic similarity between the first German case and these variants of concern. So, on a more positive note, why does the genomic tracing with networks appear to work so well? Well, if you think about it, we have a serial interval, that is the time of infection from one patient to the next, of about 4 to 5 days. And the viral mutation rate is similar. It's one mutation about every 2 weeks. So that means after a chain of about 3 infections, we can expect on average 1 genomic mutation. So quite often the new mutation links with the new patient being infected. And that's why you can use phylogenetic network tracing for identifying hotspots of where infections are happening. There is a caution, however, that this phylogenomic tracing might be easier at the beginning of an outbreak rather than when there are millions of people infected, and then you would need large databases indeed in order efficiently to trace where someone got their infection from. So that concludes my lecture. I just want to give a few acknowledgements to my colleagues. So first of all, of course, the operators of the GISAID database, without whom all this would not be possible. Then Mike Pivnenko here in Cambridge, who helped with the statistics, as did Tobias Polzin and Arne Röhl in Germany with the network analysis. I also received advice from Maureen Miller, who used to work in the Wuhan Virology Institute, from Richard Thomas, who is interested in the trafficking of animals, Shuichi Matsumura in Japan, who is also an expert on simulations, and Chris Wang, who has provided valuable advice on this talk. And most of all, I'd like to thank my mentor, Professor Colin Renfrew, who started up the archaeogenetics group with his foresight in Cambridge archaeology just over 20 years ago. Finally, here are the references on which my presentation are based, and at the end you will also find a web link where you can find our data, our analysis results, and free software, all available for downloading if you wish to conduct research on this coronavirus yourselves. Thank you very much for your attention.